So, hi everybody. Really, I'm very amazed with the results that family have achieved with effort and work. This is awesome, and it's a pleasure to be here and work with all of you. So in the next minutes, we are going to try to make easy genetics. And I will switch to Spanish to be easier to all of you, families and other professionals. Estamos acostumbrados a hablar sobre...
When we talk about rare genetic diseases, we're used to talking genetics, but many of us hadn't heard of anything about genetics until a first diagnosed befell the family. Probably you will hear about very technical aspects in future presentations, but to deal with those, maybe some of you could use some Genetics 101. So what is a genetic disease? What is a minority disease? What is a syndrome? We will try to answer these questions in the next few minutes. When we talk about genetics in general, we need to understand we each have a gene code, a guidebook, an instructions book for the body to form physical structures and work the way it is supposed to work. Adequate growth, neural development, defenses, blood pressure, blood sugar. If there are impairments in the function of these elements, we are led to believe there can be an error in the code, in the genetic code in this case, predisposing individuals to diseases, dysfunctions, and disorders. This is a cell where all the genetic information is summarized. For an easy comparison, we have DNA, which is like a big, compact encyclopedia containing tons of information. There are two types of DNA, mitochondrial and nuclear. We will focus on nuclear DNA because we're dealing with gene CTNNB1 located in the nuclear genome. Going back to the idea that our DNA is a large encyclopedia, we see a full list of chromosomes, which would be the different volumes or books in the encyclopedia containing information cells need available so that they can read it whenever they need it. As humans, we have 23 pairs of chromosomes, half from the mother, half from the biological father. We all have this genetic data system coming from both parents, and then we create further genetic information which may differ from the one we inherit from the mother and the father. Gene CTNNB1 is located in chromosome 3. This is something we will see later in genetic studies. When we read reports on where genes are located, we need to maintain this idea in mind. If we take one chromosome or one volume of this big encyclopedia and we unzip it, so to speak, we ultimately see the double helix in DNA, which sounds very sci-fi, but we can actually read it. All these nucleotides, all these letters are our gene code, and there are techniques available to read even these nucleotides, these letters that lead us to identify and diagnose different genetic diseases. Genes would be the chapters in each volume within the encyclopedia. Genes are instructions to perform the functions cells need to undertake. There are 25 to 27,000, depending on the sources, genomes, and only 15,000 are known to us today. So although we make progress every year, there are still more than 10,000 genes to decode. Just knowing that they exist and that there can be different diseases associated to them can be mind-boggling. Even genes have different components. I'm sure that if you've read any genetic report or file, you must have read the words exon and intron For years, we thought the exon was the important part of the gene because it's an encoding area, whereas the intron had no value whatsoever. But today, we know that both components are important as introns participate in the regulation of these elements. For better understanding, we can seek genetic upsets or alterations. By size, they can be large or small. And depending on the size of impairment, the technique used will vary. As part of punctual smaller alterations at a nucleotide level, there are different types. Some are synonymous and they do not change the protein or the amino acid. Others are missense or nonsense, which means different types of genetic alterations. And within CTNNB1 gene, usually there are punctual variations in nucleotides. A single letter or several letters altered involve an alteration in the protein function, leading to symptoms. And sometimes there can be genetic alterations in certain areas of the gene that entail no problem whatsoever. So the different genetic studies out there will depend on the size, depending on the assessment made in clinical evaluation. Also, many families may be wondering that when genetic studies are conducted, not usually from the start, and these take place in genetic services or neuropediatrics units. Health centers, pediatricians, and other specialists need to be aware of the possibility of referring parents to this sort of test if the child exhibits symptoms. If they take too long, diagnosis can be very late, and as you probably know, it may take months or years to get a true genetic diagnosis because the information came in too late or genetic studies were not available at the time of the diagnosis, or because new genes have been now decoded, leading to more accurate diagnosis. When alterations are large, we talk about karyotypes. For smaller nucleotides at a chromosome level, we run the array CGH study, and when we want to study specific genes or read the genetic code, there are different tests. As we said, there are 25 to 27,000 genes, and if we suspect a specific gene, we can study it directly, but this is difficult because many genes can lead to overlapping diseases or misleading clues. So studying genes one by one is very difficult. Usually we pick panels of several genes that can lead to the same symptoms, and in other areas we do an exoma, which is a study of all known genes. As for how genes are inherited, well, there are dominant, recessive, and sexual chromosomes that we will not deal with today. CTNNB1 is a dominant gene. Since we have two copies in our gene code, only one of the copies needs to be altered for a patient to exhibit symptoms. In recessive genes, both copies of the gen, or both members of the pair, should be affected. If we're only carriers and had only one recessive gene affected, we would transmit the disease without being affected by it. If we go over to inheritance, which is an inevitable word in genetics, we find the following. When we find a genetic alteration in a patient, we go back to the parents. Although we say that one copy of the genetic information comes from the father and the other one from the mother, we need to determine where the genetic alteration comes from. It can be inherited, but from either the mother or the father. At times, both parents are normal, and therefore the genetic alteration happened within the patient itself. In other words, there are no genetic alterations in the parents, and there is no need for further studies to determine whether other siblings could be affected. When someone has a genetic impairment and they transfer their copies to their descendants, there's a 50% risk of descendants inheriting the trait. Many families can have a child with a genetic alteration, and they're always concerned whether the next child could suffer the same disease. In that case, the rest of the family needs to be subject to a study to ascertain the risk of the same alteration happening all over again. If parents do not have any altered genes, the likelihood of the child inheriting the disease is under 1 or 1.2%. And you will wonder, how come it's not 0? Well, if a genetic alteration could happen once, it can also happen twice. And even if the risk is very low, during pregnancy, direct studies can be done in chorial biopsy to make sure the new baby is not affected with the same disease as their sibling. However, if either parent has a genetic alteration, the techniques used are different. If we want to determine the likelihood of inheritance, we can consider a pre-gestation diagnosis and pre-implantation. In other words, the mother is intervened and eggs are extracted as well as sperm from the father. These are joined in the lab. Embryos are studied to make sure they don't have any genetic alterations before they're implanted in the mother's uterus. In the case of families with genetic alterations. I just put these notes together to explain genetics in a nutshell.